Hello, everyone. Welcome to today's webinar. I'm Jillian Kemsley, a senior editor at CNEN, and I am moderating today's event. This webinar is titled Small but Bright, Microcolumn Size Exclusion Chromatography, Multi-Angle Light Scattering, Adds Light Scattering to Ultra-High Performance Size Exclusion Chromatography for Absolute Molecular Weight and Size. It is being sponsored by Wyatt Technology. CNEN works with sponsors to identify topics of interest and value to CNEN's audience and that are consistent with CNEN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget at the bottom of the screen or type your query into the Q&A box. If you are disconnected during the webcast, please log in again according to the instructions you received earlier. You are encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I will pose as many as time permits. Please note that CNEN does not endorse any company, products, or services that may be mentioned in the webinars, and that each webinar will be archived at CNEN online after the live webcast. The presentation today is sponsored by Wyatt Technology. Wyatt manufactures light scattering instrumentation and develops software for determining absolute molar mass, size, charge, and interactions of macromolecules and nanoparticles in solution. During the presentation, we will hear from Michelle Chen, Wyatt's Director of Analytical Services. She oversees new application development, sample analysis services, technical sales support, and instrument quality control. She has a PhD in chemical engineering from Yale University, where her doctoral work focused on the development of high-speed and high-efficiency liquid chromatography of biopolymers. I will now hand over to Michelle.
Thank you, Jillian, and good morning, everyone. In today's webinar, I'm going to introduce a new MALS detector with extremely low dispersion volume, suitable to use after UHPLC-based size exclusion chromatography. Size exclusion chromatography separates polymers based on their hydrodynamic volumes. It is an important chromatography tool for estimating molecular weight, its distribution, and for detecting aggregation in biopharmaceutical products. The development in SEC for the past few decades has been very slow or even stagnant at times until the recent introduction of UHP-SEC by Waters Corporation. UHP-SEC stands for Ultra High Performance Size Exclusion Chromatography. It is also known as UHPLC-SEC or SE-UHPLC or APC, Advanced Polymer Chromatography for Synthetic Polymers. All those acronyms refer to the UHPLC-based size exclusion chromatography using coated silica beads from 1.7 to 2.5 micron as the separation particles. Because of much reduced particle size and reduced dispersion in the UHPLC chromatograph, the resolution is enhanced significantly. And because of the greater resolution, we can now use much higher flow velocities to achieve faster separation. The peaks are sharper now, so we can use detectors to achieve higher sensitivity, even with very low sample quantity. The mobile phase consumption is also reduced. So overall, the productivity of a size exclusion chromatography separation is improved tremendously. Like SEC, when we use UHP-SEC alone with a concentration detector, we can only estimate the molecular weight based on the sample's elution volume, like those three proteins shown here, and then refer to the column calibration curve created by a series of known molecular weight standards. When we use the column calibration technique, we have to make a number of assumptions. We have to assume the sample of interest has the same conformation and density as the standards. we have to assume there's no column interaction, the chromatography is reproducible. When one or more of those assumptions fail, especially in the case of UHP-SEC, then the estimated molecular weights will carry large error. Let's take a closer look at those three proteins. According to elution volume, protein 1 should have slightly higher molecular weight because it's slightly earlier elution volume. Protein 3, eluted much later, should have a much lower molecular weight. Now I'm going to plot molecular weights measured by online static light scattering instrument as those thick, flat lines across the peaks. Now we realize protein 1 actually has a lower molecular weight than protein 2, only slightly though, 146 versus 152 kilodalton. Protein 3 also has a very similar molecular weight, 150 kD as the molecular weight. So even though they have very similar molecular weights, but they eluted very differently, we're going to propose some possible causes later in the webinar. So now we probably realize light scattering is really critical in order to measure the correct molecular weight in UHP-SEC. So I'm going to give a very brief overview of the two light scattering techniques used for online biophysical characterization. The first is the static light scattering or multi-angle light scattering, also known as MALS or SLS. It measures time averaged scattered intensity at different angles. That scattered intensity is proportional to the product of molecular weight and concentration as shown by the equation on the right. So if we measure concentration online as well, then we will be able to measure the molecular weight based on first principle without the need for making assumptions or no molecular weight standards. If the polymer is larger than 10 nanometer, we can also measure the scattered intensity as a function of different angles to calculate the root mean square radius, RG. So we can get both molecular weight and RG from a MALS measurement. The second light scattering technique is dynamic light scattering, or DLS, QALS. It measures the scattered intensity fluctuation as a function of time in very short time period. It directly measures translational diffusion coefficient, and then we can use Stokes-Einstein equation on the right to calculate hydrodynamic radius. So we can use both detectors for the SEC system. For a comprehensive SEC-MALS system, it should have a UV detector, the MALS with embedded dynamic light scattering, and the differential refract index detector at the same wavelength as the light scattering detector. As we discussed in the previous slide, scattered intensity is proportional to both molecular weight and concentration. So in order to measure the molecular weight, we also need to measure the concentration, either from the UV detector, if we know the extinction coefficient, or from the RI detector. The corresponding constant in RI detector is called DNDC. And DNDC is quite constant for most of the proteins, unlike extinction coefficient can vary greatly from one protein to the other. So if we have those light scattering detectors together with the concentration detectors for UHP-SEC, then we can extend its capability. Not only will we be able to measure the molecular weight in the mobile phase without making any assumptions in the chromatography properties of polymer shape, we can actually reveal the shape of the polymer if we can measure both molecular weight from online MALS and RH from DLS. We can also detect, characterize, and quantify protein aggregates and fragments. Because UHP-SEC has much higher resolution, it may resolve new peaks from the SEC peak, and those detectors will help us identify those new peaks. Also assess the heterogeneity of molecular weight across those peaks. And it will also allow us to characterize protein conjugates and copolymers by their composition and stoichiometry. Now, if we use the standard MALS detector for UHP-SEC, we would encounter some challenges. Let's take a look here. This is the SEC peak from a column with 5 micron particle size. And the peak volume is 440 microliter, defined as the peak width at 5 sigma, or 4.4% of the peak height from the baseline. The flow cell volume for a typical MALS detector like our Helios or Trios has overall close to 70 microliter volume, including the cell volume, the manifold volume, and some tubing volume. So 67 microliter versus 440 microliter is still rather insignificant, less than 20%. That's why the MALS detectors are compatible with the standard SEC separation. But now if we use UHP-SEC, the peak volume is drastically reduced, in this case 40 microliter, even smaller than the flow cell volume. So making the current MALS detector incompatible with the UHP-SEC separation. If we're still going to use the standard MALS and standard RI detectors for UHP-SEC separation, we will get traces similar to what's shown here. The UV is in green, light scattering in red, and RI in blue. We can see when the UV peak travels to the light scattering detector, it gets broadened visibly. And when it transfers to the RI detector, there's a tremendous dispersion. The fragment peak visible in the UV trace is completely buried in the tail region of the RI detector. So the resolution will work very hard to achieve from the UHP-SEC separation got diminished by the standard detectors. To overcome those challenges, our R&D scientists worked very hard for the past 2 years and came up with this new optical bench. It features a flow cell with 10 to 15 times reduction in volume, 20 to 25 times reduction in broadening or dispersion. They have also redesigned the re-engineered optics and detectors to minimize the stray light and also to maintain its high sensitivity as our other MALS detectors. It also has an option for DLS, for simultaneous MALS and DLS measurements. It also has a built-in in situ ultrasonic flow cell cleaning device called COMET. COMET allows us to automatically clean the flow cell after each sequence run. Now with this optical bench, we're ready to introduce our MicroDAWN instrument, the one and only MALS detector specifically designed for UHPLC-based separation. We call our solution the µSEC-MALS. µSEC-MALS refers to the use of MicroDAWN with embedded DLS and the low dispersion RI detector called UT-REX by using those detectors after a UHPLC-based SEC separation. So now we can compare the results from MicroDAWN UT-REX with the standard detectors. So we tested different samples. In this case, it's a protein by 2 sets of detectors, UV MicroDAWN UT-REX in red, UV standard MALS, standard DRI detector in blue. So let's start from the left graph, which have the UV traces. The dispersion volume defined as full width half maximum here are the same because it's from the same detector, 44 microliter. When the peak transfers to the light scattering detector, MicroDAWN saw 1 microliter increase in dispersion, but the standard MALS detector saw 11 microliter, or a 25% increase in dispersion. And when they further migrate to the DRI detector, UT-REX saw an increase of only 3 microliter increase. The standard DRI saw 50 microliter increase, almost close to 100% increase in dispersion. So I think MicroDAWN and UT-REX dispersion is indeed much less than the standard detectors. The example from a mixture of polystyrenes on APC column told us the same story. So same MicroDAWN and UT-REX traces are in red. The standard MALS and standard DRI traces are in blue. So from the left, we can see the dispersion from MicroDAWN is 90 microliter, and from MALS is 114 microliter. And when those peaks migrate to the downstream RI detector, UT-REX saw 3 microliter increase. The standard DRI detector saw a 48 microliter increase. So consistent from what we just saw on a protein example. Now we can also compare the molecular weights from those two sets of detectors. Here is an example of IgG and a fragment. If we use the standard MALS and DRI detectors, the dispersion between those two peaks are pretty visible, or the resolution is quite reduced comparing to the resolution from the UV detector. When the dispersion is excessive, Ben Browning correction feature in our Astra software cannot do a very effective job. So we see unreasonable molecular weight trend across those two peaks with the standard detectors. Now, if we use MicroDAWN and UT-REX, then we can perform the Ben Browning correction properly, and we see reasonable molecular weight trends across the peaks with correct molecular weights. So from this, we can confirm that MicroDAWN and UT-REX maintain the resolution from UHP-SEC. They provide accurate molecular weight and size information. And now we can perform proper Ben Browning correction in Astra software. And most of the key online MALS applications can be readily transferred to the HPLC platform. Now, in addition to the low dispersion volume, we would also like to learn more about MicroDAWN's characteristics. So we tested at different flow rates with a model protein. We tested at 0.075 ml per minute to 0.15 and 2.3. Even though the elution volume changed slightly with different flow rates, but molecular weights from MicroDAWN are consistent at different flow rates. We actually also tested in a separate experiment even a higher flow rate, 0.5 ml per minute. We did not see any surprises. So we're pretty confident that MicroDAWN works well at a wide flow rate range, including the low flow rates. Sensitivity and reproducibility are important aspects of any detector. So we tested here a mixture of proteins at different loadings from 0.4 microgram to 6.4 microgram. And molecular weights are plotted across those peaks. As you can see, they're very consistent. At the lowest loading, 0.4 microgram, the light scattering signal shown here is still very strong. For a signal-to-noise ratio of 10, we can actually reduce the loading amount 10 times less at 40 nanogram. So microdon is among the most sensitive MALS detector in the world. We also tabulate the results of the molecular weight for those two proteins here. And as we can see, the standard deviation, the relative standard deviation is less than 1%, indicating those detectors are highly reproducible and robust. We all know light scattering detector is very sensitive to large aggregates because light scattering sensitivity increases with molecular weight. But how about for the low molecular weight end? So let's take a look at this IgG sample. We see a dimer peak at 311 kilodalton, and then 157 for the monomer. We also notice there's a peak eluding very close to the total inclusion volume, and we actually can also measure the molecular weight to be 0.36 kilodalton or 360 dalton, consistent with the molecular weight of a disaccharide. And we know in the sample there is a disaccharide present. So microdon provides high sensitivity also over a wide dynamic range. Now we are happy with the microdon's performance. It not only has low dispersion volume, but also is sensitive, robust, reproducible, works for a wide molecular weight and flow rate range. So now we're ready to test some real-world samples and applications. All the data presented for the rest of the webinar were collected under this experimental condition. We used the water's acuity UHPLC system. For proteins, we used the BEH column, either 15 or 30 centimeter in length. For polymers, we used the APC-XT columns. The UV detector is also from waters. The MALS, obviously, it's from our microdon. And the DRI, low dispersion volume DRI, is Optilab UT-REX at the same wavelength as the microdon. First, I'm going to share some applications and data for proteins and biotherapeutics. The first example is four monoclonal antibodies. We saw a similar chromatogram earlier. They're supposed to be monomers, but they elute at very different times. Microdon revealed all four antibodies have very similar molecular weight from 145 to 155 kilodalton. So they're indeed monomeric. But their elution times or volumes are very different. Could it be due to their different hydrodynamic volumes, due to different shapes? Well, we can check that with our online dynamic light scattering instrument. The embedded DLS inside the microdon produces very high quality autocoloration curve shown here. And actually, this online DLS can measure the RH value of IgG down to 20 microgram per milliliter. We find those four IgGs have very similar hydrodynamic radii as well, from 5.1 to 5.2 nanometer. So those four IgG monomers have very similar molecular weight, very similar hydrodynamic radii. Their different elution volume must be due to column interaction. Actually, this happens quite often with UHP-SEC, the column interaction, probably because the limited surface chemistry of the stationary phase available on the market currently. And this emphasized the importance of having microdon and dynamic light scattering for correct molecular weight and size measurement. The second example is a mixture of proteins. By standard SEC, we find 2 peaks. When we also use the UHP-SEC to analyze the same sample, we seem to also detect 2 minor peaks, which can be seen much better if we zoom in. Now we're going to rename those peaks as peaks 1, 2, 3, and 4. And the 2 peaks from SEC would correspond to peaks 2 and 3. Now we ask ourselves, can we identify those new peaks by retention volume? Can we identify them by MALS and UV alone without RI? Well, without further analysis, I can already answer the second question. Well, we cannot use UV alone with the light scattering detector, because if we use UV alone, we will need to know the extinction coefficient. And we do not know the identification of those new peaks, so we would not know the extinction coefficient, and we would not be able to measure the concentration from UV detector. So we will need the DRI detector to measure the concentration, so light scattering can measure the molecular weight. But if we do have UV detector online, then we can calculate the extinction coefficient by using both UV and RI. We tabulate the molecular weights and extinction coefficients from those 2 systems here, and they actually give very similar results for peaks 2 and 3. Peak 2 has a molecular weight of 145 kilodalton, extinction coefficient about 1.5 for 1 milligram per milliliter. Peak 3 molecular weight is 67.6 kilodalton, extinction coefficient about 0.67. Even though those 2 peaks differ in their molecular weight by a factor of 2, but they're not simply monomer and dimer because they have very different extinction coefficients. Now, to answer the second question, actually we already answered the second question. To answer the first question, I will move to the next slide, because in here I also plot the molecular weight against the elution volume, as you can see. And now we can tell peak 1, even it eluted earlier, actually has a lower molecular weight, 134 compared to 146 of peak 2. So now we can answer the first question. No, we won't be able to get the correct molecular weight based on retention volume. And we already discussed that RI is needed for molecular weight determination. And by the addition of dynamic light scattering, we can also get the hydrodynamic radius. So by using MALS DLS, RI, and UV, we can identify those new peaks by their molecular weight, RH, and extinction coefficient. Protein stability is directly related to the efficacy and safety of a therapeutic protein. It is often studied under different stress conditions, stressed by temperature, light, pH, and other mechanical forces. In this example, we have a very stressed IgG sample, and we can use the light scattering detector together with UV and RI to identify those peaks molecular weight and extinction coefficient. We identified the intact IgG, FAB2 fragment, FC fragment, and FV fragment. Now, if we shift our focus to the peaks eluded before the intact IgG peak, we see some aggregates. Let's zoom in and also overlay with the trace from a blank injection. Injection was just a mobile phase, so we can be certain all those peaks are from the sample, not from the system, as the case here. So we can use microdon to measure the molecular weight, the first peak molecular weight is from 1 million to several millions, likely aggregate particles. Now let's focus on the soluble aggregates and zoom in further. And we can see their molecular weights vary from 188 kilodalton to 307 kilodalton, representing different forms of the aggregation. One of the most powerful applications for MALS, UV, and RI is its ability to characterize those protein conjugates by measuring the composition and stoichiometry. So this application can be easily transferred to the UHPLC platform. And we have an example of glycoprotein here, which is from a virus surface. We can resolve multiple forms of this glycoprotein, and then we can use UDAWN, UV, and RI to measure the total glycoprotein molecular weight and plot it as the top lines here. We can also tease out the molecular weight from protein and plot it as the middle lines here, molecular weight from glycans plotted as the bottom lines here. And by looking at those molecular weights, we conclude the main peak eluting at 2.7 milliliter is a monomer, and the peaks before the monomer are a dimer, trimer, or even a hint of tetramer. So now we can actually use UHP-SEC to separate protein conjugates as well. We can also use µSEC-MALS to compare biosimilars and their innovators. Before I'm going to discuss the different biophysical and analytical evidence we can gain from µSEC-MALS, I would like to bring your attention to the dimer peak here. You probably notice there's a curvature in the molecular weight across the dimer peak. Unlike the flat molecular weights across the peaks we saw earlier. But this curvature usually is the telltale sign of self-association nature of the protein. In this case, because the peak eluded with a concentration gradient, so there's a different mixture of protein oligomers in the peak. So now let's see the evidence we can gain from µSEC-MALS for those biosimilars. We can measure the molecular weight. We can quantify the percentage of monomer, aggregates, and fragments. We can determine extinction coefficient and hydrodynamic radius of each peak. We can also detect signs for potential self-association. So µSEC-MALS can provide us evidence of biosimilarity with greater confidence. The last bioapplication is quite unique for UHP-SEC. But we often use size exclusion chromatography to measure the fractions from a prep scale HPLC, either for purification or manufacturing of a protein. The SEC analysis time typically is 15 minutes or longer, limiting its application to offline analysis. Now, if we use UHP-SEC, then the analysis time can be greatly reduced, making it possible for potential at-line process monitoring. Let's take a look at the example here. We took 3 fractions of a large-scale separation, and then use the UHP-SEC to analyze those fractions. Instead of 15 minutes, we were able to do it within 3-4 minutes. And the addition of the microdome can help us identify the aggregates, the fragments, and possibly impurities. Now I'm going to summarize all the different µSEC-MALS applications I discussed earlier. So we can use the system to measure absolute molecular weight, size, and extinction coefficient, even there's column interaction. We can also assess the heterogeneity or homogeneity of molecular weight across the peak, looking for signs for impurity and self-association. We can also characterize protein conjugates by using the microDAWN with UV and RI. µSEC-MALS system facilitate the transfer of the method from standard SEC to UHPLC-based SEC. And also because of its higher resolution from the UHP-SEC separation, we can offer stronger biophysical and analytical evidence for biosimilars. It may also open potential application for process monitoring. And now I'm going to share some data from APC, Advanced Polymer Chromatography, for some synthetic polymers. This is the APC separation of the Polydisperse Polystyrene, NBS-706. And just like some of our customers have used the standard MALS and DRI for APC, we did see a decent peak with reasonable molecular weight across the peak. Because the sample is polydispersed, so molecular weight is not flat, but rather has a pretty steep slope here. We also used the microDAWN and UT-REX on the same system and same column. And we noticed the peak is slightly sharper and the molecular weight is a little bit wider. The same sample was also analyzed by a GPC system with 2 linear columns and using the standard MALS and DRI detectors. So the results are pretty comparable, but APC speed is much faster. Now we're going to tabulate those results for the 3 systems. We tabulate here the number average molecular weight, weight average molecular weight, polydispersity, which is defined as the ratio of weight to number average molecular weights, and the Z average loop mean square radius, or Z. As we can tell, those results are very similar, especially for the weight average molecular weight and Z average loop mean square radius. But there's some differences within the PDI, so I'm going to discuss those. First, we can tell that GPC gave the highest polydispersity. Usually when the number average molecular weight is the same, a higher PDI would indicate a better separation. And this is mainly due to the fact that we use the 2 linear columns for GPC, and APC, we just use the 2 columns with pretty large pore size, so the molecular weight range is not as wide as the GPC columns. So that's why we got a high PDI from GPC. The second difference I want to bring your attention to is the PDI from the standard detectors and from microDAWN UT-REX. microDAWN UT-REX has a higher PDI, and this is due to the low dispersion, so then you can get a more complete separation by using those detectors comparing to the use of the standard detectors. So APC can also benefit from the use of microDAWN and UT-REX. High molecular weight polymers are very sensitive to shearing degradation. So we wonder, does APC cause shearing degradation? So we tested with a high molecular weight polystyrene sample, 1.6 million gram per mole. And actually we did not see any appreciable degradation. But the flow rate is pretty moderate, 0.5 mil per minute. I know many APC users use much higher flow rate, 1 or even higher mil per minute. So we're going to test the higher flow rates after this webinar, and only microDAWN can tell us whether there's shearing degradation or not. Before I conclude, I also would like to share a couple questions asked by our customers. Does µSEC-MALS give the same results as SEC-MALS? Well, if the sample is not affected by the different chromatography separations, then they should give the same result. The second question is, can microDAWN and UT-REX be used with the standard SEC column? Well, in principle, it is a yes, but we usually don't recommend it because those detectors, microDAWN and UT-REX, would have a much higher back pressure, and it can potentially shock the column or clogging the system. But if you use a very low flow rate, like 0.2 mil per minute or lower, then the back pressure is not a problem. microDAWN does perform better at the low flow rate. So I hope I have convinced you that microDAWN and UT-REX are very helpful for UHP-SEC separation. They actually preserve the speed and resolution from UHP-SEC, and they can provide additional information. In addition to molecular weight, they can also measure RG, RH, extinction coefficient, and conformation. And all key online MALS applications can be transferred to this UHPLC platform. And actually, the addition of the light scattering detector is going to help the method transfer from the standard SEC to UHPLC-based SEC. The high speed and wealth of information may open new doors in process monitoring and other areas we yet know, and hopefully our customers will tell us down the road. And I would also encourage you to surf our website to learn more about light scattering theories, white products, application notes, and search our bibliography for peer-reviewed articles. You can also listen to on-demand webinars on our website. Now I would like to acknowledge my colleagues for making those wonderful detectors and also for collecting those beautiful data used in today's webinar. And also I would like to thank you for listening. I'm now ready for questions.
All right. Thank you very much, Michelle. We have several questions already, so we'll get right to it. The first question maybe goes to something you mentioned right at the end there. We have someone asking, it seems that microDAWN is a lot more sensitive compared to the Helios 2. Is there a way to use microDAWN in standard LC runs?
Thank you for the question. Actually, you may, but the concern that I had and mentioned in the webinar is that the back pressure is rather high at 0.5 mil per minute. The microDAWN can generate about 20-25 bars, and UT-REX may generate 15 to 20 bars. So the high back pressure makes the system risky for shocking the column or causing a blockage. So that's why I think you need to be very careful when you use those detectors with regular SEC columns.
Okay, and can you give us a sense of what are the limits of detection and dynamic range of the system?
Okay, I think this question may divide into several parameters. For molecular weight, the molecular weight range we can measure is from several hundred daltons, like the disaccharide, and up to few millions, depending on the conformation, because this microDAWN has 3 light scattering angles, so it can measure up to several millions. Now, for the radius gyration, the range is 10 to 50 nanometer. And for hydrodynamic radius, it's from 0.5 nanometer to 30 nanometer at 0.3 mil per minute. Actually, a lot of those specifications can be found on our website, product, MALS, and microDAWN.
And can you actually use the system to calculate radius of gyration?
Yes. It has 3 different angles at 45, 90, and 135. So it can measure the radius gyration from 10 to 50 nanometer.
Okay. And we have another question asking how many detectors and at which angles?
3 at 45, 90, and 135.
Okay. All right. Can you describe how an analyst would set up a molecular confirmation experiment using the QUELS module in online SEC?
Okay, so the QUELS is actually embedded inside the microDAWN. So you just set up the microDAWN after the UV and maybe before the UT-REX detector, and you will simultaneously get molecular weight and hydrodynamic radius. So from the correlation between molecular weight and hydrodynamic radius, you can get information on the shape and conformation.
Okay, we've had a couple questions, I think, for low molecular weights. Could you provide some numbers for sensitivity for low molecular weights? And they're saying, low for them is 1 to 3 kilodaltons. And what would be the recommended loading amount for micro SEC models?
Okay, so because light scattering intensity is proportional to molecular weight and concentration, so if I give one number for a particular molecular weight, we can always calculate the detection limit for the other molecular weight. So actually, again, you can find on our website the sensitivity specification for BSA is 70 nanogram loading on a 15 centimeter BEH column. So 70 nanogram for BSA at 67 kilodalton roughly is about, let's say, 35 nanogram for IgG, or 50 nanogram for 100 kD protein. So now if it's for 1K, it's 100 times less in molecular weight, which means we need 100 times more in concentration. So for a 1 kD peptide, the minimum loading would be 5 microgram.
Okay. And another question here. Could you please elaborate on the calculation of the extinction coefficient from RI determined concentration?
Okay. So because we have both concentration detectors online and their concentration of mass need to conserve. So if we know the concentration from one detector, then we can calculate the unknown parameter for UV will be extinction coefficient from the UV detector. So I mentioned earlier the proportional constant for the RI detector is called DNDC. And DNDC is quite consistent for different molecular weights. It's dominated by the amino acid peptide bond rather than the side chains. So DNDC for most proteins is the constant. And so by using RI, we will be able to know the eluted concentration or the mass in the peak. And so we can calculate the extinction coefficient if we don't have a detector online.
Okay, we now have a couple of questions about flow rates. On the high end, what is the maximum flow rate that the microdetectors can handle?
The microDAWN can handle up to 1,000 psi or 70 bars. So that may correspond to flow rate at 1 ml per minute for some systems or 2-3 ml per minute for other systems. So I think the parameter we should go by is the pressure it can handle. It's up to 1,000 psi.
And on the other end, are there any concerns using flow rates less than 0.1 milliliter per minute on an acuity, such as on an acuity M class? And do you have any suggestions for UPLC columns that work well for protein separations?
Okay, so the first question, yes, we have tested flow rate below 0.1 mil per minute. Actually, earlier I mentioned some flow rates study, and we tested at 0.075 mil per minute, and we saw still very good data. There's no anomaly at this low flow rate. And second question, sorry, I don't remember the second part of the question.
Sorry, I cleared it off my screen, but I think it was, do you have any suggestions for columns that work particularly well for protein separation?
Okay, so for our studies, we used mostly the Waters BH columns. I know there are some other manufacturers also offer those 2 micron particles as the stationary phase now. But for most of our studies, we use the Waters BH columns, and they seem to work well.
Okay, and someone would like me to clarify, is microdon more sensitive than Helios?
I think in terms of the concentration, sensitivity, they are comparable. They're the same. The minimum detectable concentration for microdon is 1 microgram per milliliter for BSA, or 0.5 microgram per milliliter for IgG for a signal-to-noise ratio of 10. And I think Helios has the same specification, and we tested, verified they all can produce a signal-to-noise ratio of at least 10 at this concentration. However, Helios, because of larger dispersion, it will broaden the peak, and the microdon can preserve that sharpness, the height of the peak. So in that regard, microdon can be considered more sensitive than Helios. But in terms of the concentration, they have comparable sensitivity.
Okay. Is the system able to detect the different folding status of a protein?
For folding, I believe you probably will use the combination of molecular weight and hydrodynamic radius. And for that, the minimal difference in hydrodynamic radius we can detect is probably 0.1 or 0.2 nanometer. So if the different folding states will produce a difference in hydrodynamic radius more than 0.2 nanometer, then yes, we will be able to detect it. Otherwise, we won't be able to confirm by the online QELs, however, online dynamic light scattering. However, maybe we will get some clues from the elution volumes. If we combine molecular weight and elution volumes, that may also tell us some information on the folding states.
All right. And do you need dN over dC ahead of time, or can it be calculated on the fly?
It may be calculated on the fly. When it's being calculated on the fly, we will have to assume whatever we inject will elute, so-called 100% mass recovery. But if you have a UV and you know the extinction coefficient, then you can also calculate the DNDC that way.
Okay, a couple more questions and then we will wrap up. I'm currently happy with my GPC-RI measurements and molecular weight without light scattering. If I transition to APC, should I revisit the need for light scattering?
I think so, because APC, as I mentioned, may cause shearing degradation, may see new peaks, and column calibration may have some abnormal behaviors, all those you would not be able to know without absolute molecular weight detector like the microdon online. So I think when you transition from GPC to APC, I would like to have a microdon to guide me and to tell me anything that may be hidden or not being revealed by the column calibration alone.
All right, and are microdon and Optilab UT-REX compatible with HFIP?
Yes, they are compatible with HFIP or most organic solvents. And actually, that's probably a good application because HFIP is very expensive. And when you transfer the method from GPC to APC, you may save significantly in solvent consumption.
All right, and lastly, one final question. Are µSEC-MALS detectors and software compatible with 21 CFR 11?
Yes, they are compatible with 21 CFR 11, and also they come with IQOQ. If you need those documents, we also provide that.
All right. Well, thank you to our audience for all of the questions. You've been a great audience. Thank you, Michelle, for your presentation. Everyone, be sure to check CNEN or CNEN Online for information on the next edition of CNEN webinars. Thank you, On24, for Technology and Production Service. And thank you, Wyatt Technology, for the sponsorship that made this interactive webcast possible. For CNEN webinars, I'm Jillian Kemsley. Goodbye.